Professor Markus Manz, the head of the Center for Hematology and Oncology, about immunotherapy in hematologic malignancies. Markus.
Well, thank you, Rolf, for the invitation, also Michael. I thought as we had Professor Ralf Bargou here this morning, then we would already have covered almost heme, but thanks for giving me the opportunity to also talk on this. And I always like to show this hematopoietic tree, saying this is a paradigmatic somatic stem cell supported organ system, and within this system there are possibilities for malignant transformation. Of course, it's the hematopoietic system, but also it's the immune system. And if we talk about immunotherapy, then we talk in this case of a system which actually should do the immune job, but where the disease is occurring within. So that's a somewhat special situation. It's almost like an infectious disease that is targeting a immune cell, as for example, HIV. Another interesting aspect is that with this system, in contrast to many other somatic supported stem cell organs, or maybe also self-renewing organs, it is possible that if a disease would occur here, then the eradication of the disease as well as the compartment where the disease comes from is possible, because temporarily you can live without it and then it can regrow. So I think there are some special aspects about diseases that originate, malignant diseases that originate from the hemato-immune system. And of course, hematologic malignancies are somewhat present in the field, also at the bigger centers, but overall, of course, these are only about 10% of the neoplasias, and within these 10%, the variability is extraordinarily high. The diseases, with exception of acute lymphoblastic leukemia, are diseases of the elderly population, with the ALLs probably particularly frequent in the young people, because the immune system is built up and all this T cell and B cell rearrangement occurs, and there's a lot of necessary genetic instability that allows then also the occurrence of potential tumor-driving alterations. But otherwise, these are mostly diseases of the elderly, and they are located in somewhat the middle of causes for tumor death here, at least in Switzerland, but that's true for a lot of countries in Europe and the Western world. So in Switzerland, the population is aging. It's expected that in 2030, there's a doubling of the population of over 65, and that means also if the age-adjusted incidence is stable, then it also would double, and if our therapies are successful, then also the prevalence will enormously go up. So it looks like this is, as with other tumors, a challenge for the future. Now, if we talk about immunotherapy, we'd like to talk about precision medicine, and I also always like to say what's the ideal of precision medicine. It's that the target is essential for the tumor, it's a driver, and if you can block it, then you have tumor elimination. Also, the target should preferentially be exclusively in or on the tumor and not in healthy tissues, saying a neoantigen, important in all this checkpoint inhibition, or the therapeutic target is on or in the tumor and in healthy tissue, but the healthy tissue is not functionally relevant, so some prostate cells or some immune cells that temporarily can be missed. Yes, as I said, the target should not be relevant for survival. Actually, up here it said that the target should not be in the healthy tumor or it should be there but functionally not relevant, which is frequently the case for proto-oncogenes that are active in development but then are silenced during adulthood as long as there's no major repair. And of course, that would guarantee a few side effects, and if you combine both, then you would have a magic bullet, as we think we have, at least for many cases of chronic myelocytic leukemia, where you have this kinase that is very specifically active and you can very selectively inhibit, and in CML, targeted therapy, not immunotherapy, causes a survival in the CML group that is now approaching the survival of the healthy population. But that, of course, is really a rarity. Now, what are the principles? I will be brief, we also heard that today. So the immune action can be T-cell mediated, antibody mediated, NK cell mediated, phagocyte mediated, or a combination thereof as the immune system works. Or there might be newer artificial intelligent designer immune mediated processes like bispecific, trispecific, multispecific antibodies, also click antibodies, and of course these connections of an antibody with a cell that makes it a particularly strong effector like CAR T cells. And then the mode of application could be either the effectors are transferred or the activators of endogenously existing effectors can be transferred, again talking mostly about checkpoint inhibition therapy. And this is depicted here once more. So with antibodies we can have complement activation, we could have direct action if it's blocking a receptor that is essential for the cell survival. We could have antibody-dependent cellular phagocytosis initiated by the connection of the antibody with the Fc receptor and then the consecutive activation. And the same is true for the NK cell dependent cellular cytotoxicity that is initiated via this Fc activation through the antibody. Then we have the T-cell activations, and that again is what mostly matters for checkpoint inhibition. You have the tumor cell, hopefully a neoantigen is presented on MHC. The T-cell receptor recognizes it specifically. It's activated via a co-stimulatory molecule, potentially a cytokine, and then if it recognizes via the specific TCR, then it induces killing. And of course, talking about checkpoints, the tumor cell as healthy cells, because that is a mechanism that has been established with, for example, chronic viral infection, that not the whole organ is eliminated. Hepatitis C infected liver cells express the ligand, so the T cells are not killing all these hepatocytes. That causes then the inactivation, and for a long time one tried to activate by giving cytokines, vaccines, etc. And now what works is basically inhibiting the inhibitors and therefore causing then the killing. This is designer immunology. Professor Bargou talked about the BiTEs, the B-specific T-cell engager, MHC independent. The TCR is not recognizing the antigen, but it's a connection between something that is associated to the TCR, and then if this is engaged and this is engaged, the antibody fragment in both directions leads to the same for T cell as the TCR activation, meaning signaling for activation and then killing, or the alternative that, again, not the MHC mediated killing is used, but a antibody fragment is brought into a cell that then signals upon ligation and the T cell is activated and leads to killing, or another designer mode of immunotherapy to conjugate something to an antibody or an antibody fragment. That could be a cytokine, it could be a chemotherapeutic, it could be something that emits irradiation and then relatively selectively brings this substance to a tumor cell and kills it. And I would like to just say one thing about this antibody-dependent cellular phagocytosis, because I think this is something that we will see in the future as an important element of therapy, is that this would be the classical activation pathway, but what we've learned is that every cell in the body expresses an antigen called CD47, which ligates to SIRP-alpha on phagocytic cells and tells the cells not to be activated, even if there's a signal 1 that says activation. So this is an inhibitory signal that's kind of an innate checkpoint for macrophages, and if one generates antibodies that are covering the binding epitope here or here, then this don't eat me signal is not transmitted to the macrophage, and the eat me signal becomes stronger, and then the cell kills the respective target cell, in this case a tumor cell. Actually also something that nature is practicing. So there are a lot of pox viruses that infect the phagocytic cells, and then they upregulate homologs of CD47 to protect them from being eaten up. And also we know it from ovarian cancer, other cancers, has been published a long time ago, that they upregulate this CD47. Actually also stem cells, hematopoietic stem cells that go in circulation and pass by a lot of phagocytic cells, they upregulate CD47 on their surface and therefore escape this scavenging of phagocytic cells. And maybe the best studied is the CD47 expression on red blood cells. If red blood cells age, they downregulate CD47 and then they are eaten up in the spleen. So important innate immune mechanism that so far is not in clinics approved, but is in phase 1 clinical trials. So I tried to talk about various aspects of immunotherapy in hemato-oncology. One is allergenic transplantation. This is the oldest type of immunotherapy, cellular immunotherapy. Of course, first the tumor is reduced, but usually not eliminated, and then cells, hematopoietic stem cells, are transplanted to reconstitute hematopoiesis, but also immune cells, active immune cells, are transferred that protect from infection, that cause GvHD. That's something we don't want, but they also cause GvHD against hematopoiesis in the context of allotransplant. In this case, we don't say GvHD because that's not nice. We say graft versus leukemia or graft versus lymphoma, and that sounds better. But in the end, it's the same thing. That's allorecognition, and then there's activation, and the threshold seems to be lower than for the classical GvHD organs like liver, intestine, and skin. But that's the basic principle, and it's clear that if you take an identical twin and transplant him or her from the respective, must be female or male with the identicals, then the remission will not last long, but if there's a little bit of a difference, then the remission lasts longer, and that shows here if you do T-cell depletion and transplant stem cells or transplant from twins, then the relapse rate are way higher, and but the GvHD rate is lower, and if there's more diversity, then also the GVL or GvHD against hematopoiesis and lymphopoiesis is starting. So that works, and it has been tested now whether in this situation and relapse it would also work. It's clear that you can either transfer donor lymphocyte transfusion, well described, but also you can, instead of doing donor lymphocyte transfusion, you can simply give checkpoint inhibitors. That has been shown here with the CTLA-4 blocking monoclonal antibodies, and this is from the New England paper, showing that there can be massive responses. In case this was Hodgkin's lymphoma, which is particularly interesting, but also in AML there have been shown responses in about 25% of this small cohort, and interestingly, all those that responded had prior GvHD, again linking this GvHD-GVL effect. Now, is there a hematologic malignancy where the checkpoint inhibition is really showing some success, and that also is published a while ago in the New England. These were 23 patients and has been repeated with a lot of other patients that were refractory upon the primary treatment, also upon the salvage treatment, and they were treated with nivolumab in this case, and the response rate, overall response rate in relapsed refractory Hodgkin's disease was very high, and now it's also in Switzerland allowed to be given in patients, don't have that slide in patients that are after high-dose salvage or after anti-CD30 therapy, brentuximab, and if there's a relapse, then they can be treated with checkpoint inhibition. But of course, this also goes now in studies in earlier phases of disease, because it has been observed that, interestingly, in classical Hodgkin's lymphoma, PD-L1, PD-L2 alterations exist in sense of disomy, polysomy, copy gain amplification, etc., that define actually is a defining feature of classical Hodgkin's lymphoma. That's also shown here, and this amplification leads to higher expression, and we know from the histologies of Hodgkin's lymphoma how this immune infiltrate is there and how few Hodgkin cells are there, and actually these cells sit there and they're inactivated because the Hodgkin's lymphoma, particularly these B cells, these transformed B cells, express these high ligands due to the activation of the gene in the tumor. Interestingly, this is close to JAK2, and if this is amplified, usually also JAK2 is amplified, which again might be a target for therapy. Are there other leukemias, lymphomas that benefit from checkpoint inhibition? A lot is trial, and a lot is still ongoing. What can be said is that it looks like virus-related lymphoma, EBV, Hep C-induced lymphomas are particularly sensitive, and that makes a lot of sense. Also, CNS lymphoma, testicular lymphoma, and primary mediastinal B-cell lymphoma are also responsive to this type of therapy. Actually, it was just approved, Keytruda was just approved in relapsed refractory PMBCL. Myeloma didn't look so promising so far, but we will see. So what's another approach? That's optimized monoclonal antibodies. There are these targets, particularly in B-cell development, that are expressed and are not expressed on the earlier cells and are not expressed in any other tissue. And of course, the classical approach has been rituximab for a long time now in clinics, has been shown in most of the randomized trials as being of benefit for those patients. Virtually every B-cell neoplasia that expresses CD20 now gets rituximab in addition to chemotherapy, and mostly the curves go not only significantly but relevantly also diverge, and it's, as I said, is used in most indications. Can we do better? That's the question, and there are many efforts to do that. This is one effort to improve that, and that is by modifying parts of the antibody to induce better ADCC and induce better direct cell death. And this is glycoengineering. It's called GA101 or glyc-art, glycosylation art, was actually developed here in Schlieren and then by an offspring of the ETH, and then was bought by Roche, and Gazyvaro carries still the name like glyc-art, and Zurich and Roche all in there, and that's used. It has been shown that it works better than MabThera in certain settings of CLL, and that was progression-free survival, but at EHA 2018 it was also shown that now in the study also the overall survival is better. There is some different dosing in these two arms. The rituximab has not got the same dosing as the GA101, and there's some discussion of whether the effect might be due to that. I'm not sure whether it ever will be solved. In vitro, clearly the glycosylated antibody shows a stronger effect, and also when you infuse it, the immune reaction that you can observe in the patient is way stronger. So the inflammatory response that you create is way higher. And of course, MabThera has a problem with its, I would say the other way around. There is the patent is running out, and it's great to have a new product that works more efficiently. Now, are there other antigens on the mature B cells that can be targeted? What you can see here is that CD20 is not expressed on plasma cells, and that is a problem because it works in all B-cell neoplasias that have a certain maturity, at least in the mature B-cell neoplasias, but it doesn't work in plasma cell neoplasias because there, with the downregulation of the surface immunoglobulin expression and the production of something soluble, this is downregulated, but there are other molecules expressed, and they can be targeted, and one of them is targeting CD38, that's called daratumumab, that has been tested in various combinations with effective therapies in multiple myeloma, particularly in the relapsed refractory setting, and this is showing 2 trials published in New England in autumn 2 years ago. And what you can see here is the expression of CD38 here very high in the myeloma cells, while it's lower on hematopoietic stem and progenitor cells, and that gives the therapeutic window. This is also a optimized antibody where the Fc part is optimized for more ADCC, more complement-dependent cytotoxicity, and the curves look like that. So there is a massive difference in this relapsed refractory situation with both a proteasome inhibitor, also with an IMiD, where the addition of the antibody and the maintenance with an antibody shows a better outcome, here defined as progression-free survival, that also lead to the approval of this drug in similar situations in Switzerland. And in February of this year, the first trial in first line was published in patients that are not going to therapy intensification and autologous transplantation. Also there's a clear difference in terms of adding daratumumab to bortezomib and melphalan and prednisone. And the hypothesis is that this anti-CD38 monoclonal antibody will make its way in first-line therapy of multiple myeloma. So bispecific antibodies I'm not going to talk about, but I'm going to talk a little bit about CAR T cells. This is kind of the story that has been told over and over again. This little girl, 2012, in relapsed refractory acute B lymphoblastic leukemia. They wanted to send her to the hospice, and the parents and friends found out that in Philadelphia there is a new type of therapy in the lab that kills CD19 positive cells, and she was going on a study there, and this shows her in autumn, coincidentally with the ASH meeting, the biggest hematology meeting in New York Times, well placed, and then 1 year later and 5 years later with her Emily Whitehead Foundation now to foster these types of therapy, and she is a survivor so far of a situation that seemed terminal, and that is how is it done? T cells from an individual are taken, genetically modified to have a B cell receptor with a signaling domain on the cell. This is this, and then if this is given back to the patient, and if this antibody that is engineered on a T cell recognizes its antigen, then the signaling domain will tell the T cell the same signal basically as if the T cell receptor would recognize this antigen, and the activation signal varies depending on what is genetically engineered here. These are the studies: relapsed refractory ALL, very good event-free survival and overall survival with this type of therapy that doesn't include chemotherapy, at least not chemotherapy that is intended to target the tumor. Sometimes people need some therapy to reduce the lymphocytic compartment, so you have homeostatic expansion of these transferred cells. So if you transfer T cells in a T cell empty individual, then there will be homeostatic expansion even without seeing an antigen to a certain level, and that is used in these type of studies. These cells can, in some cases, survive for a very long time, and usually it's also those individuals where you can detect those transferred cells for a while, that these are the individuals that do respond. Based on these trials, this was the first approval of a CAR T cell therapy in relapsed refractory ALL. The product is called Kymriah, and it's a product by Novartis. And in the US, that's approved for younger patients with B-cell precursor acute lymphoblastic leukemia. It's interesting, if these cells are transfused and the tumor volume is high, then the activation of those cells is so strong that is an enormous cytokine release syndrome. It's almost like, someone getting the flu and lying like this in bed, and one important mediator for that is IL-6, which is released, and not by the T cells, but is released by the system, and that can be antagonized with an antibody called tocilizumab, which was co-approved in this special situation to reduce the side effects without obviously reducing the efficacy of those T cells. Now it's clear that this will not stick to acute lymphoblastic leukemia, but will move on to lymphomas, and there are two bigger studies published again in the New England Journal of Medicine from two different companies using this in relapsed refractory non-Hodgkin's lymphoma, and also there in very advanced situations, the response rates and survival rates are relatively astonishingly high. And based on this data, also these two indications are now approved by the FDA, one in October 2017 and the other in May 2018 for diffuse large B-cell lymphoma, primary mediastinal B-cell lymphoma. Actually, only Yescarta is having this label, high-grade B-cell lymphoma, and diffuse large B-cell lymphoma from follicular lymphoma, so transformation. I said what are the differences, at least between these two products. It is the co-stimulatory domain, so this is the difference. This is where the efficacy, the stimulation, the release, but possibly also the survival is influenced by, and what that means on the long run for those CAR T cells, we don't know. There are subtle differences. Now, is this limited to only CD19 as a target? No, of course you could take any target. I think this morning there were also some solid tumors mentioned, like prostate cancer with BCMA, thyroid cancer could be a target here. It's shown for refractory myeloma, and what you can see here, when B-cell maturation antigen is targeted by such a CAR, at least in this patient and then subsequent in other patients, but the good ones are usually shown. You can see that applying this therapy leads to a massive reduction in PET-CT and also a reduction in the light chain. So there will be other diseases that can be targeted by CAR T cells. One problem is that you need to take the T cells from the individual, modify them, give them back. If this individual has been under a long immunosuppressive therapy, then the T cells are not going to work so well. So wouldn't it be better to have kind of off-the-shelf CAR T cells that you can use and do not need to rely on the endogenous product? Also from a manufacturing point of view, that would be better. And there are, again, efforts to do that. This is one example that was published in Science, basically as an anecdote where a baby had a relapse and after allotransplant didn't have own T cells, and the donor T cells were also not available. And in this case, third-party T cells were used, and they, of course, bury the risk of a allogeneic response. So there was there was another engineering, but I'm not talking about it, but two-type engineering, one bringing in the CAR, and the other eliminating the endogenous TCR of these donor T cells so that the GVHD would not occur. And these were transferred, and there was a response in this leukemia. There is multiple ways to engineer that in addition, saying that you could put two CARs in, and only if both CARs find their ligand, then there would be full activation. So if you only have two signaling domains that signal, then you would get a response. Or you could do it in a way that you have two CARs in there, and if there's a normal cell with an antigen that's also expressed on the tumor, but the normal cell has in addition another antigen, it could give an inhibitory signal. So the way to combine this is multiple, and the aim is to increase the selectivity of those cells. We also do that actually together with Michael Weller here. We have a consortia called Immunocure, where we generate monoclonal bispecific CAR T cells, not for lymphatic neoplasias, but for myeloid neoplasias and also neoplasias of stem cells, and of course for neoplasias in the brain, but we don't have anything in the clinics yet. We are still at the bench and in the animal house, respectively. Now, I talked about this eat-me control, this innate checkpoint inhibition. I just wanted to show that there are multiple phase 1, mostly studies ongoing, and we don't have data yet, but we will have at least in autumn at the ESMO Immuno-Oncology meeting, we'll have the head of 47 Inc., who will talk about their progress there. So I think hematology has the luck to be kind of a front runner in multiple ways to use the immune system to actually fight again parts of the immune system, and I'm very excited about that. And of course, I'm happy to take questions if I'm still allowed to, because I already took too much time. Thank you very much.
I'm sure we have time for one, two questions, if there are any. No, thank you.